someone might ask, okay, so each of these clusters, can we name them based on a type of cell? And if we get some external information about what are typical cell marker genes, then we might be able to plot these on this map. So now let's make a new file here. I would say marker analysis. So I'm gonna define a new function that takes the RNA and this function plots classic immune marker genes on the RNA map. And so there are some classic markers defined, such as CD3D and CD3E. So these are T cell marker genes. And it has also subsets, CD4 and CD8A. So these are T cell subset markers. And we have MS4A1, which is B cell marker gene. These are NK cell marker gene. Sorry, Z. Monocyte. All right. These are known marker genes for PBMC immune cell types. Now I'm gonna use a list comprehension to filter marker genes that actually exist in the dataset. So if G in RNA high variable genes dot variable names. Now let's import and plot. This time we use the color to be available markers. and we're going to return available marker. Okay, let's run this. So let's take a look at the results. Okay, so if we just look at it at first glance, these two are really look-alike, as all the left blob and right blob are dark purple and the middle one is kind of yellow. And so based on our definition here, yeah, both of them belong to the same marker gene. So that's good. And then we have, which else is similar? Yeah, this guy and this guy looks very similar. They're both light in this section only, and maybe a little bit green here, then again a little bit green here. Yeah, so let's check NKG. Yeah, these two are also NK cell markers. This guy looks very different because it only has this cluster light up and the rest are dark, which we don't see in anything else. MS4A, yeah, this is a B cell marker, and I'm guessing 1, 2, 3, these are also not really that similar with each other. This guy has the left blob very light, this has something in the middle, and this is like this cluster and this cluster. Yeah, so these could be separately indicating different markers as they are only by themselves. So this is telling me our RNA features are extremely informative. Like the RNA features genuinely encode cell type identity, and the information we need to predict proteins actually exists in this dataset. Now, the last thing I want to do in this EDA is to calculate correlation between marker RNA genes and matching proteins. Create another function here. Marker correlations. So I'm going to have RNA that is pre-processed and proteins that are already having log transformed. First, I'm going to make a variable called RNA express, XPR, and I'm going to make a data frame. Out of this RNA and data matrix that we have, so RNA p.x, this expression, the one that we once converted it to an array, we have to do it again here because it's a space matrix. And else we're gonna keep it if it wasn't sparse matrix. I'm gonna keep the index as RNA observation names. And I'm going to keep columns as variables. And so again, rows are going to be cells and columns are going to be genes. Again, we're going to find common cells. Between proteins and RNAs. So intersection protein log dot index. I'm gonna keep only the common cells in both RNA and protein. So we're going to define gene protein pairs, the ones that we expect to have relationships, like we already know them, such as CD4, CD4, and with CD4, CD8A and CD8, CD14 with itself. All right. Now I'm gonna go over these pairs in a loop. Gene protein keyword in gene protein pairs. And I'm gonna fix each pair, and then I'm gonna loop again through every matching protein that we have in our protein log. And then I'm going to calculate the correlation between each of these genes with every protein. All right. And so first, I need to check if gene exists in RNA expression columns. If it wasn't, then we're going to go over the next pair. We're going to skip through this if the gene is missing. And then I'm going to find the matching proteins. And make a list comprehension. P for every P in protein log columns. If protein keyword that we're looping over from here may match a protein column containing what's in P, right? So this is going to find us all the protein whose name contains the protein keyword. Why do I have this? Oh wait, in, okay. And so once we have this list, we're going to loop over it for proteins in matching protein. I'm going to define the correlation RNA expression. We're going to pass the gene here. And we're gonna choose the method Spearman. It's gonna calculate Spearman correlation between RNA and protein. And I have to keep them all in, let's say, a list. We define here results, and then results dot append. Can make a dictionary that has the gene name and protein. And we have Spearman correlation. And let's print them too so we can see. Gonna be like gene RNA vs protein. This is going to give us 3 decimal places. And then I'm going to come out of the loop, both of them, and then I'm going to return the data frame version of results. That's results. Okay. Alright, now let's go ahead and see what we're gonna get. Okay. So these two are having negative numbers, which means they are unrelated and not a good prediction pair. These two are moderately difficult to predict, like predicting the protein from the RNA. But this guy, CD14, is probably easy to be predicted from CD14 RNA. So these were some random gene protein pairs. Maybe if we want to see, like, if we can make sense of what we saw in the UMaps earlier, maybe if I change some of this to, let's say, so we had CD16, maybe if I add NKG7, because that's the gene associated with CD16, and we have, say, CD19, the gene associated with it is MS4A1. So let's see how this would turn out. I'm seeing the strongest signal here is this guy, just like a little bit different from this, but when we look at the UMaps, CD19 definitely had the strongest signal of taking the whole 6 cluster lighting up here. And so that is perfectly consistent with what we already discovered so far. And this number totally makes sense. And from biology, we know that CD19 is a marker for B cells. Also, when we look at this, they're moderately related. And CD16 here is lighting up cluster 4 and then 8, which is pretty consistent with cell markers. Yeah, at least we have this here. And for CD19, we have MS4A, which is lighting up on the cluster 6. So I would say these are pretty consistent, and the correlation also reflects that. But I think our code found some false matches too. Here, I would want this to require the keyword to be followed by underscore. So like CD8 only matches CD8 underscore, not CD80 underscore, or like CD8 6 underscore. So I'm gonna make another case here, plus. And I think that will fix this bug. All right, let's add these and commit. Commit these files. Add EDA, marker, gene, and protein overlay analysis. So that wraps up the exploratory data analysis. We confirmed that our RNA data captures clear cell type structure our protein markers align with RNA clusters, and we have a good sense of which proteins will be easier or harder to predict. Next up, we will move into pre-processing, and then after that, the modeling itself. Hope you had fun coding with me, and see you in the next video. Bye!